Hello, I'm Archana Hari from UMBC, and today I'll be talking about computing, analyzing, and visualizing genome-scale metabolic flux networks using a tool called Fluxer. Genome-scale metabolic models comprise all of the chemical reactions occurring within the cell or organism. These models can be simulated using a constraint-based flux balance analysis to compute rates of reactions called fluxes and how they contribute to cell behavior in various metabolic phenotypes. It is, however, challenging to study and make inferences from the mathematical solutions that FBA provides. Visualizing the fluxes through all the reactions will alleviate this problem, and this was our motivation to develop Fluxer, which is now a user-friendly and open-access web application. Users can load any genome-scale metabolic model in the SBML format or use any of the over 80 readily available models. Once a file or model is loaded, graph computation is performed where the SBML reaction and metabolite namespace is compared to reaction and metabolite information tables housed within Fluxer to identify the cofactor metabolites, get crosslink information about these entities, etc. Next, FBA is performed using CobraPy, and the results are converted to graphical representations using various graph algorithms. There are three major graph representations that Fluxer generates. These are the spanning tree, the k-shortest paths, and the complete graph. Each of these graphs are bipartite representations with the green nodes representing reactions and the yellow nodes representing metabolites. The edges on these graphs indicate the movement of flux or the direction of flux either towards or away from the root node. In the case of the spanning tree and the complete graph, the color of the edge also indicates whether flux is moving towards the root node or away from the root node. In the example over here, we can see the spanning tree for or Plasmodium falciparum with a zoomed in version of what was found as the most important pathways that happen to be the central metabolic reactions such as glycolysis or pentose phosphate. The next graph that Fluxer is able to generate are the k-shortest paths graph. And these are, as the name indicates, the k-shortest paths between the user-selected source and target node, which could be reactions or metabolites or both. Users can select whether they want the shortest paths to be generated in terms of length or edge weight. And the edge weight is a customizable combination of the reaction flux, the metabolite stoichiometric coefficient, and also the molecular weight. Each path has a color associated with it that can be referred to from the key shown below. The graph over here at the bottom illustrates how the inclusion of cofactor metabolites such as hydrogen ion or ADP can result in biologically infeasible shortcuts. Users will thus find it useful to exclude these cofactor metabolites from the graph computations, and Fluxer allows them to do so. Thus users can select whether they want cofactor metabolites or even reactions carrying no flux or zero flux to be excluded from the graphs. And these settings are applicable to all three graphs, the shortest paths, the spanning tree, and the complete graphs. The shortest paths graph finds important applications in metabolic engineering. So the last graph that Fluxer is able to compute is the complete graph, and this contains all of the reaction metabolite nodes and all of the relationships between them such that nodes may be duplicated as well. The complete graph is shown in the force and radial layouts over here, but they're also available in the tree and endogram layouts as for the other graphs. In the force layout, users can view global level clustering such as in the example over here, where reaction and metabolite nodes are clustering based on their cellular localization. Finally, Fluxer is able to simulate reaction knockouts. In the example over here, the reaction cytochrome oxidase is knocked out in E. coli, and as expected, the fermented pathways become active, shown by the red edges over here. All of these graph visualizations can be exported in the SVG or PNG format. The nodes can be clicked on for more information and also to view the nearest neighbor's mini graph. Additionally, the model can be viewed in the SPML layout if that is defined per the SPML layout's extension within the model file. Thus, in conclusion, Fluxer is able to perform FPA and even reaction knockouts and compute genome-scale metabolic representations based on spanning trees, k-shortest paths, and complete graphs. In the future, we want to expand Fluxer so that users can customize FPA in terms of objective function reaction and also media formulation. We also want to implement new graph layouts so that various metabolic phenotypes can be visually analyzed better. I'd like to thank the members of the Logo Lab and the students and faculty at UMBC for the support and especially in their help in testing the web application. We thank ISMB 2020 and ISCB for the fellowship award. For more information, you can visit the web application, which also has a informative tutorial page. Alternatively, you could view the paper cited over here for Fluxer, which was recently published in Nucleic Acids Research web server issue. Thank you.